Protein engineering. So, it is the process for developing the protein which having the desired function, structure and specificity. It is termed as protein engineering. Now, this is done by taking two approaches. First is the rational approach, or we can say rational design, and second one is irrational, or we can say directed evolution design. Now, in rational design, we are having the protein structure and function, and in that we are changing something according to our requirement. So the mutation is planned and it is known as rational design. This is the difference between two protein design. First one is the rational protein design. So the knowledge based engineering of protein which desired characteristic. So here we having the protein, the knowledge about the protein structure and function, and from that we are getting our desired characteristic protein. Now starting in that design or modeling, so it is totally computer based or computer aided. Next, generate required changes in cloned DNA construct. Now here we are cloning the protein. It means we are replacing the segment or part of protein with the another and express the protein. Now expressing the protein into the DNA, so that a particular segment which are applied into the protein particles is multiplying the new protein, or we can say our requirement of protein is done successfully by applying that particular segment in the protein structure. Now purify that protein and then assess the protein for desired changes. Next, we are talking about the irrational protein design. So, it is the high throughput protein engineering. In this, we are not knowing anything about the protein. We are blindly running behind the desire to get running behind the getting desired protein structure and functional. So, in this, generate library of DNA constructs. Here we are having the library of DNAs. It means in library there are so many books. Meanwhile, in this design we having so many DNA constructs and some bioinformatics. Bioinformatics is the obtain, store and interpretation of biological data. So bioinformatics having the biological data. Now this express all constructs. So we having the library of DNA and we are expressing, and this protein is screened and designed for characteristic, and ultimately we get the DNA construct for the larger scale expression. Now rational design change rational design is making change in the gene of the protein. The production of protein molecule is changed by the site direction or site specific mutagenesis. Here we are doing the mutagenesis process to alter the protein structure. Alter it means changing the structure of protein, and by this we are getting the desired protein. Next is objective of this protein engineering. So enzyme, antibody, hormone, receptor protein, etc. are protein class chosen for engineering. So in this method, or we can say in this process, we choose any one of them, and mainly the enzyme is mostly used for the protein engineering process to improve the usefulness and efficient protein. The protein engineering has the objective of very first is increase the stability of enzyme. So here we are increasing the stability, increase the catalytic efficacy of enzyme, introduce the new active site and new catalytical activity in enzyme. So in enzyme, if we are adding new active site, so the bounding capacity of enzyme is broader, or we can say it wide the range of binding capacity. Also, it alter the pH dependency of the enzyme. It improve the thermostability and modify the substrate specificity of enzyme. Next is understand the structural and functional relationship of the enzyme for making the prediction of action. So here we are understanding the structure and functional of the enzyme. Next is producing the tailor-made enzyme in the large quantity by increasing the expression of structural genes. Tailor-made, it means what? We need enzyme like this. So here we are preparing the enzyme. એઝ આપણે જેમ કપડાં સીવડાવવા જઈએ તો એને ટેલર કહીએ, એવી જ રીતે અહીંયા આપણે we are producing the enzyme according to our requirement in the larger quantities, producing the hybrid enzyme and making the isolation and purification of the enzyme easily, and the hormone resistance to attack by the antibodies or the stomach enzyme. So this kind of the enzyme are produced and ultimately used in the therapy, detection, analyzation, so many